Okay, this is hopefully the last video here to finish up the biology air practice test. And we are on, I think this is question 62 or 63. But we're going to try to wrap up here, so I'm going to try to rush through because I've got about 8 or so questions to go. Looking at this question, this has got to do with our egg lab again. We've had a couple questions like this already. These are supposed to be red blood cells, and they are in different solutions. So inside the red blood cells is basically like a 50-50 mixture of water and ions from salts. On the outside, though, there's different concentrations. So like on the left, we see it's mostly ions. In the middle one, we see mostly water. And on the right, we see it's about the same amount of water and salt outside as it is inside. So what we're seeing is the salts, because they're charged, they do not go through the membrane very well, really hardly at all. So the salts are kind of stuck on one side of the membrane, but what can change is that water can move through the membrane. Water can go through. Water is a small molecule. It's polar, but it's not charged. It does travel through the membrane. This is called osmosis. Osmosis is the traveling of water through the membrane. And just like anything else, osmosis is a type of diffusion. So water will go from a high concentration to low. So we're looking for water molecules are the big blue ones. And we're looking for where's the high concentration, where's the low concentration. That's going to tell us the direction of motion. So there's more water here, and there's less water outside. So in this one, water will move out. This one has more water outside, so water will move in. And this one is pretty balanced, so this will actually go in a little bit of both directions. And so there's the motion of the water molecules, and not surprisingly, we should be able to figure these ones out. If the water is going to move out of the cell, this cell will shrivel. If the water moves into the cell, this cell will swell up and potentially burst. And this one, since it's kind of balanced going in and out, we should see no real effect. It should keep its normal size. So there's our 6-part answer to that question based on osmosis. Alright, next, this is a sort of a complicated thing, but the idea here is that DNA strands, the two opposite strands, are held together by hydrogen bonds, right? Hydrogen bonds between each strand. And then when the DNA gets copied, we kind of unzip those hydrogen bonds, right? So those hydrogen bonds are fairly weak, and they can be broken by heating the DNA. So if you have two chains, two opposing sides of a strand, and they match every base pair, an A and a T, a C and a G, they've all got hydrogen bonds connecting the two together, which means that you have to raise the temperature fairly significantly to get those things to unzip. Now, if you compare two similar species, their protein chains are going to be kind of matching. Not their protein chains, I'm sorry. Their DNA chains are going to be kind of matching, but they're not going to match perfectly. So maybe instead of having an A and a T, maybe there's an A and a C. And those two don't match right, and that means they're not going to hydrogen bond. So what we see in this chart are groups of species and how much cooler. This is actually how much it says their separation temperature, how different they are. This is how much lower the separation temperature is for other species, for different species of DNA, because of those hydrogen bonds being missing, because the pairs aren't lined up right. So because they don't line up well, we lose some of the hydrogen bonds. That means that we can break the DNA apart at a lower temperature. So the numbers that are closest to 0 have the fewest differences, and the numbers that are biggest have the most differences in their DNA chains. Because there's, again, the more differences means more breaks in hydrogen bonds. So our question, part A, says which pair of species is most closely related. Again, they're the ones that have the smallest temperature difference, and that would be species 2 and 4. Part B, you're supposed to pick statements that sort of describe what's happening here. And I believe B, similar temperatures are needed to separate DNA from species that share more similarities. Again, the more close they are, the more hydrogen bonds they get. You're going to need warmer temperatures. So that's B. D, I think, is the other one. Species that have diverged from a common ancestor more recently will share more complementary base pairs. Base pairs is kind of off the screen, but that's what that one's saying. So B and D are the right answers there. Next up, we did this one in class. This is a this is incomplete dominance. So the idea here is if we have homozygous on one side, we can get black chicken feathers. If we have homozygous with the different allele, we can get white chicken feathers. And heterozygous means we would get blue chicken feathers. That's one white allele and one black allele. We would get blue feathers. So the way a student could test this is by having one parent that is purebred black, one parent that is purebred white, and we would see blue offspring. A is the answer here, because if this offspring comes from these two parents, they should be heterozygous. So if they're correct, the heterozygous should come out blue, and so A should tell us if this is what we're looking for, if this is a correct analysis of the type of inheritance that's happening. So A, black feathered parent, white feathered parent should make offspring with blue feathers because they'd all be heterozygous. Next up, chief cells. These are in glands inside your stomach. They make large amounts of enzymes. Enzymes are a type of protein. They're produced and secreted from the chief cells to help in digestion. When we say secretion, we mean they get released from the cell. So which cell structure is abundant in these cells? Enzymes are proteins, right? So protein production and secretion is part of the job of the endoplasmic reticulum. It helps to produce, it helps to develop the proteins into their mature form. It helps to fold them, and it can label them for transport. And it also helps them then to leave the cell. If that's the job of the protein, is to leave the cell, the endoplasmic reticulum can help with that job. So it's very important in producing and sort of transporting the proteins where they need to go. So since enzymes here are proteins, the ER, the endoplasmic reticulum, is the best answer there. How is chemosynthesis different from photosynthesis? Photo means light, chemo means chemical. So chemosynthesis occurs in environments without sunlight. We use chemicals from the environment instead of light to make our food. That's a chemosynthetic organism instead of a photosynthetic organism. So that's B. Alright, what's happening here is we are looking at a graph of how a deer population changes when a new section of territory is acquired. So this red line represents where new territory will be acquired, and we're looking at the carrying capacity. So the deer right here, that's the carrying capacity that we see. This is how much room, this is how many deer could live in this park, and this red line represents, again, where we're going to see a new section of park being added. So what we expect to see during B is the population size will get a chance to increase because we're getting more room. And the carrying capacity during that time will also increase. So we're going to see a bump up here as more deer are born during this time. And then after the 10-year period is up, they're going to stop the survey, and we're going to make a guess as to what we're going to see after that. And probably after 10 years, we probably are going to have, the population's probably going to be where it needs to be. So the carrying capacity during time C is probably going to be at equilibrium. There's nothing changing here other than they're going to stop doing a survey. So once it's up, it should stay at that level during time C. So it should be at equilibrium. So those are our answers there. We're looking at bird preference in worm size. What kind of size do these different worm do these different birds prefer to eat? So species 1, remember these are frequency curves, so mostly species 1 likes these guys here that are about this size, but they'd eat them a little smaller, and they'd also eat them a little bigger. And then species 2, again, that's the preferred size right in the middle there, but they would also eat them a little smaller or a little bigger, and same thing for 3. So there are some overlaps in what these birds will eat, but what we're looking at here is a range of worm size, and if there's a bunch of variety of worms available, these guys should not have to resort to competing with each other very much because their favorite worm size is right here, and the other birds don't really like that size. Their favorite worm size is here, the other birds don't really like that size, and their favorite size is here, and the other birds don't really like that size. So again, if there's a nice range of sizes here, these birds shouldn't have a lot of competition, and I believe that's going to be our answer here. So A, no competition. No, that's not true. There's some competition. At least 3 different species of worms are eaten. We don't know how many species. Selection pressure, result in a worm population with only small and large worms. No, probably not. Less competition will be observed if a range of different sized worms is available. That was our answer. Alright, and our last question, which seems to be unanswerable for the first part. We did this in class. What we're looking at is the environment changes so that the brown frogs do better than the green frogs because of predators. They're more camouflaged. So what you should be able to do, which I don't think you can do, is put some dots over here, some green and brown dots. And what you're going to see in the new population after a few generations is there should be basically more brown dots and less green dots. Green frogs are getting eaten more, brown frogs are surviving more, so we should see more brown and less green in the new population. The driving force behind that is natural selection. Natural selection because some of these guys are not surviving, and it's for a reason. It's because of their better traits. They have an adaptation that lets them survive better in the new environment. Alright, and this is the end of our test.